Yeah, so yeah, thanks for coming. This will be, I mean, we hope this is just like a chill rundown through our model and what it does and all the basics and how to set up some simple simulations. So this is not going to be very much in depth into the code or into modifying it. It's mostly just how to set up simulations, how to generate landscapes to simulate like in simple topologies in with habitats, and that in some of the fitness costs like fertility reduction, mortality increases, and stuff like that. And before getting started, uh, there's two ways to run the webinar content. The one most people will be acquainted with is just downloading the whole thing and installing the dependencies. I hope you could all install the dependencies here as it is stated in the webinar's address. You might increase the font size just in case. In this, if anyone's running it this way, just needs that person just needs to install these two dependencies and this one, and download the whole GitHub repository from the address. And that would be just opening it as usual in our studio. That would be fine. The other way to run it is the way I'm going to be using here, because this one ensures that I'm running exactly what we uploaded to GitHub, is with Docker. This one just briefly is just virtualizing a machine so that we can all get the same packages, all the same things in the same way, so that we make sure that we can all run it exactly the same. Some notes before starting. If you're running it from GitHub, there's a folder that will hold all the simulation output. It will be sims out. So just don't place anything inside that folder that you need, because it wipes it automatically whenever the simulation runs. So that's it should only wipe CSV files, but just in case. For the ones running it in Docker, wherever you bind the container, it will create the same folder. Just don't place anything important there. And it won't save any changes you make to the code. So if you want to take notes or anything, make sure you save them somewhere else, because when you close the virtual machine, it will delete it. And one final note, if anyone has any questions, please feel free. I think it's way more dynamic and interesting if we just go with the questions right away instead of just waiting till the end. And I think that's it in terms of how to set up everything. I don't know if there are any questions or comments so far. Otherwise, we can get started with this end. So for the ones to get started, let's open the folder with the repository, or the ones in Docker, just launch the image and use the MGDrivE as a user and webinar as a password. I'm going to be running it in Chrome, but it should also run in Firefox. And let's see if that's running. Let's go to the introduction to MGDrivE. So, as most of you might be already aware, MGDrivE is composed, it's constructed in a modular way, at least version 1. I'm going to be talking about version 1 for now. It's constructed in a modular way, where there's three main sections, three main modules. The first one is the biology one, the life history, in which, as John mentioned, we extended Penny's framework to contain genotypes. So mosquitoes go through their egg, larvae, and pupae stages, and we keep track of their genotypes in metapopulations, let's say in larval habitats. So they all mix together in a larval habitat, and we keep track of the counts with the breakdown of each genotype. And they transition through these three phases. These three are not very exciting in terms of the code, but it's it adds a delay in terms of the development. And and at this point, they are not differentiated from between males and females. Once they pupate, we the code selects the amount of mosquitoes that it's going to assign as males and females, and then they pupate and they go through their adult stages. At that point, if they are in the same habitat, they might mate, and the females, when they emerge, they pick a male to mate with, and they store the genotype. Well, they store their own genotype and the genotype of the male with which they mated. So that way, we know which combination of genotypes we will need so that they lay eggs. And males are a bit simpler in this regard. They just emerge and mate as they need. They don't do much. It's not a very exciting life. And from that point, well, they all have a given mortality, both in the aquatics. Well, the carrying capacity is set at the aquatic stages. So that's important in this case for the population dynamics. And there's a constant mortality both in the aquatic habitats and the adult stages, so they start dying out depending on their species. And I believe that's it in terms of the life history. I can't remember if there's anything else. Depending on the genotypes, we will be adding some mortalities, some selective mortalities and some other costs, but that's a bit more specific in the next phases. So yeah, I don't know if there's questions so far or everything looking good. Let's see. Arguably the most important module of MGDrivE is the inheritance cube. This is where we pick as I was mentioning, the genotypes of the female and the male. Every female is storing this combination, and according to this combination, in the z-axis we store how many eggs of each genotype combination should we expect them to lay. So, for example, if the female is a wild type WW and the male is wild type, we usually would expect them all to be wild type. But this is the structure that can't handle those combinations, and it can be plugged in and plugged off and swapped. So, for example, we can build a traditional link drive, split drive, even Wolbachia, which is not technically a gene drive. But this is all possible due to this structure, and most of the work we do deals with this kind of analysis. And there's a couple of examples here, but we're going to just go through it. This is also the structure that allows us to add fitness costs or fitness advantages. So, for example, as all the structures in the code are vectors, we can add fertility costs to each genotype combination. So, for example, if we are releasing a gene drive that for suppression that acts on the homozygous combination of genotypes, we can add a fitness cost to the fertility of the female that only activates if that female is homozygous to the gene drive. Or, for example, if for some reason our gene drive causes the males not to be fit for mating, we can add it there. Let's say if it was, it penalizes every copy of the allele of the gene drive, we can have a cost for WH, H being the transgene, and then another cost for the HH, and that would be handled by this one. We can also have sex ratio at emergence. So if for some reason our drives biases the sex ratio, we can do it there. And some other things like the pupation success, lifespan reduction. And this one is a bit more contrived, so I'll skip it for the Wolbachia. But These are the main ones that can be done out of the gate. There's a couple of more that can be implemented, but it's specific to the case. Some are a bit more complicated. And in B2, there's way more things we can do, but this one's strictly version 1. And finally, we arrive at our landscape. Our landscape, it's fairly simple. It's a meta-populations model in which I tend to imagine it as a network of habitats. So for example, each one of these nodes would contain all the necessary things, all the things mosquitoes need to develop and emerge and mate and whatnot. And based on the distance and their migration probabilities and some of the biology, we calculate how probable it is for a mosquito to, from one node, to move to another. This happens usually on a daily time step. And yeah, I think that's pretty much it. It can be calculated for any kind of network. We will be doing a square topology, well, a ring topology, just to demonstrate it. But the limit on how large it can be, it's usually computational cost. So that's about it. And it can handle arbitrary landscapes on flat and spherical topology. So that's pretty neat. And it's usually calculated with some of these migration kernels that transform the distance into a probability. And then finally, we can also handle batch migration. This is for cases like ADs that tend to lay eggs in tires or jump into cars or whatnot. That one handles those processes. So we can add probabilities of mosquitoes just jumping into a car and being driven away, so that we can calculate the probabilities of fixation in regions in which we don't want them to be. And I think that's it in terms of the most basic introduction to the structure of MGDrivE 1. I don't know if there's any questions so far, or everything looking good. From this.
I have a question.
Yep.
So for the spatial network, you said that the probabilities of movement are calculated if you give it some structure. Is there a way to manually change any of those probabilities between nodes?
Yeah, definitely. This one, we usually don't even do it straight in MGDrivE. We have other tools we use. So for example, you could actually just lay some nodes and assign them the probabilities you want in a matrix. The way MGDrivE handles it is you store a matrix where the rows are the source, the node, the source node of the mosquito, and the columns are the destination. As long as the rows sum to 1, you're good. You can feed it whatever you need. The only constraint is that they sum to 1. I think it might even work if they don't, but mosquitoes would be disappearing, so that wouldn't make a whole lot of sense. But yeah, you can feed it arbitrary matrices, and we will be using one that's somewhat arbitrary, well, somewhat artificial, but yeah, definitely.
Thanks.
I had a question.
Yeah, for sure.
About the normalization that you mentioned here for each node. Does this mean that the overall probability to disperse is the same for each node, but
not necessarily. So for example, let's say the node 30 for some reason was extremely attractive to mosquitoes. We could make, well, they have it's not calculated as diffusion, I guess. It's calculated in let me backtrack a bit. So for example, in this case, let's say the node 30 was extremely attractive. There's a probability that the mosquito stays there. And then if it doesn't stay, then it can migrate somewhere else. So if this one was extremely attractive, we can make them just stay there for either stay there or bias the migration matrix to make them more attractive to make it more attractive for mosquitoes to move in but not out. It doesn't have to be symmetric. That's the.
But I mean, the emigration rate from a node, is it higher for nodes that are got many neighbors than nodes that are more isolated, or is it normalized so that it's the same kind of emigration rates, but you just have more different options to where you go to? Does that make sense? The question?
Yeah. I can see that. I think within MGDrivE, well, I think it depends a bit on how the matrix is calculated. So if there were more nodes, let's say there were a lot of nodes close to one, it would, if they are at the same distance, given no other constraints, if it's calculated the same, it would just have more options to integrate to the other ones than if you only have two. It would have more options. The rate would be the same, but it would just have more options. But there's no reason why, like if you want to make a matrix, let's say different, it would still hold as long as it's a well-formed matrix that sums to one. And some other modifiers can be added so that mosquitoes stay in more complicated things. So yeah, I don't know if that completely answers the question.
Yeah, no, I think, yeah, I get that. Yeah, I mean, I was wondering about how the kind of default was set, but yeah.
The default behavior would be that. The default behavior would be that it would spread the emigration rates homogeneously, but it can be modified as long as, actually, we usually just handle the matrices in MGDrivE, another package that I'm putting together. But yeah, definitely.
Okay, thank you.
Yep, no problem.
Okay, Nigel here from the other side of the planet. I'm kind of curious about, are you thinking about the differential movement of males and females in this sort of?
Yeah, sorry, I forgot to mention that. Yeah, we do have structures for male and female movement. I don't think we have used them, but it is there. That's usually just how I forget. But there's if needed, there's a different movement matrix for males than females. I don't think we have used it, but it is there. Yep.
Okay. No. I just thought, do we actually understand the biology of the difference in movement between males and females? I mean, we kind of have a handle on that with Aedes mosquitoes, but I don't know about Anopheles in Africa, but in our part of the world, we have no idea. So we would be running on sort of a generalized model.
Yeah, exactly. That's usually why we don't use it, because we have not a lot of grasp on a lot of the details. But yeah,
it's cool. Are you feeding in information from mark release for capture experiments? Males and females, and can that be built into that system? I guess it could, because it's iterative.
Yeah, it can. We have used some data just to calibrate these kinds of kernels of the how far we expect them to be found at given a certain release. It can be more complicated, but the way we would sell it, I guess, would be if there's a reason why you find that you have a better way to describe the movement, by all means, you can just feed the matrix, and we have done it before. Like we at some point, Tomas, one of our postdocs, generated a different migration simulation, and we just took that matrix, plugged it in, and it behaved more reasonable than the default parameters. But yeah, it depends on how much information you have on your study site, I guess.
Okay, great, thanks.
Yeah, no problem.
Hector, we had a raised hand from Mateo.
Oh yeah, so sorry. Please go ahead.
Yes, no, don't worry. But maybe it is a silly question, but I'm wondering if the network topology can evolve over time.
Yeah. I don't, well, in MGDrivE 1, we didn't code it to handle that. It wouldn't be impossible to modify it because as it runs in a discrete time step, you could theoretically load a different matrix over time and just load the migration at one point, then do some modification on the network, then load it again. But I don't think we incorporated that just because of computational speed. That would be a bit cumbersome. Well, a bit slow. But let's say my most concrete answer would be I don't think it's built-in, but there's nothing in the equations that would make it impossible. Not that I can think of, because I remember we discussed it at some point. That being said, version 2 should be able to do that. So maybe that would be something to think of in terms of that other version.
Thanks, Hector and John.
Yeah, no problem. I don't know if there is any other question. Oh yeah, Summit.
All right, so I guess based on that, so right now there is no, the movement is not based on any kind of density feedback, right? So for example, if population becomes too dense, mosquitoes or insects move out.
No, not in this version at least. It could be handled in version 2, but in this case they just start. They just get to the habitat cap in terms of the aquatic, and that's it.
Okay.
I don't know if there's any remaining questions. Otherwise, we can jump into the code. Okay.
Okay, I'm happy to do a last question. Look, in our part of the world, we're dealing with a malaria vector that has a coastal distribution, just a one-dimensional distribution. And we have some mark release. We capture, data mainly about females. I'm curious as to how adaptable this model is to moving from a two-dimensional system to a one-dimensional system.
Oh, it's extremely easy. I mean, so for example, in that case, and I think it's extremely similar to what we're gonna do in one of the examples. In that case, instead of your matrix connecting to adjacent nodes in 2 dimensions, you just connect let's say node 1 to node 2, node 2 to node 3, and so on. And that way you just generate a line, and the code will still run. So that wouldn't be a problem at all.
Okay, great. Thanks.
Awesome. Yeah, no problem. So yeah, let's get started with the code. So I'm gonna jump into, let me see, should have numbered this, but we're gonna go through the basic simulations first. The first examples are gonna be a Mendelian inheritance cube. This is just as a benchmark and pretty good way to get started. And then an IIT Wolbachia release, just to test a bit more. More interesting track, I guess. So let's head to the demos folder here. And let's do first the Mendelian no cost example. So we have it here running. And so I'm going to run a bit through the code. The first part, this part, let me increase the font size, might be as well. This part is not really that important for the simulations. This is mostly boilerplate code so that we can all run the same thing if we are in Docker, or if we are in a repository copy. This is mostly just boilerplate to set all the folders, all the directories, all the structures, loading all the constants, and that's it. We're gonna go ahead and run it just in case. And that's pretty much it on that one. Then we start with the interesting parts. As I was mentioning, so we're going to start with a pretty simple landscape. So it's just one node. As you can see here, we're setting up a matrix that contains just one row and one column, and it has one in it, so that all mosquitoes, so that we generate a node and no mosquitoes can get out, so they will just stay in the same population. And we're gonna set up an adult population to it. We're gonna assign it an adult population. This adult population I'm defining in the constants file. So here we can see that I've already set up some simulation constants, the simulation time to 10 years, the population to 2,000 mosquitoes. These ones are a bit more for another example, and some of the parameters for Aedes aegypti, Gambi, and Colucci. And so we're gonna go back here. We're gonna set up the bioparameters for Aedes aegypti in this case, just for the demo. In this case, these parameters are we are taking, I don't remember the sources for these ones, but we are assuming they are taking 5 days on average in egg state, 6 days in larvae, 4 in pupae, their daily fertility 20, the number of eggs they would lay, the population growth as this one, and the adult daily mortality as 0.09. I don't remember how long of a lifespan that would be in adults. I think it should be like 2 weeks or 3, but in any case, those can all be changed. So we set up the bioparameters and we load this Mendelian cube. These are defined in MGDrivE. Some of them are defined in MGDrivE. We have some examples here, link drive, split drive, Radle, and all of these ones. In this case, we just load it in and we set up our releases. We are gonna say that these releases are set up as vectors in which we set up at which time we're gonna start them, how many are gonna take place, and at which release interval, and how many mosquitoes we will be releasing. We have helper functions to do them at a regular interval, but it can be anything. You could make a release at one point, then release the next day, then 5 days later, 1 month later, it doesn't really matter. And we set up where these releases are gonna take place in the queue. And just run it. Finally, we run our simulation. It'll take a couple of seconds. And we run the whole code in this case. We will get these 2 plots. These are the defaults we have within MGDrivE to visualize our results. In this case, we're having a bunch of releases in a bunch of male releases, and that's why we see these spikes only in the male population. The genotype we're releasing is lowercase a, homozygous lowercase a, and the females reflected a bit later with mating, and it stays constant throughout just because we are adding no fitness cost to the introduced gene, so it just stays there constant. And I believe that's it for this example. This one's a bit, well, the first one is always a bit confusing, but yeah, that's it. So for example, let's say we wanted to do only one release, we could change it here. Yeah, we have one release, or if we wanted to have 10, but at a different release interval, say every 30 days. Just why not? There we have it. These results, the demo can store the plots automatically if we set this constant to true, plot to file, but I'm leaving it as false just because I think in that case it doesn't show it in the demo. I don't know if there's questions about this one or everything looking good. Okay, we can move to a more interesting one. Mendelian with cost. So for this one, we're going to do the same thing, but this time we are setting a life reduction, well, a life mortality, an adult mortality increase. So we're going to reduce the lifespan of our released transgenic mosquitoes by a 60%. And we're going to do the same releases. So we would expect it to fade out instead of just staying constant. And this is what we see. We're releasing the same, I think we're releasing the same amount, but this time as we set up this fitness cost, it is dying out slowly. And the way we set it up in MGDrivE into the inheritance cube is, we know because of the specifications we saw here, that omega is the genotype specific modifier on adult mortality. So as we are setting this omega to this genotype to have that fitness cost, it's gonna drive it out of the population slowly. If we don't set it, it will be defaulted to 1, but if we set it, it will change. So for example, let's say we wanted it to be smaller, we can do this, and the curve should be linear. Or let's set it to be higher, and let's say we wanted it to be with a heterozygous condition. This would be weird, but the dynamics, the package handles it. It would be a bit weird, but anyways, just for the demonstration. And this is the way most of the fitness costs work. Some of them are vectors, so most of them are just the combination, well, the genotypic combination of the mosquito. Some others, like the tau, need the male and the females. So I don't think we're going to use those in this example. But in that case, we can just contact us by email, because those are a bit more contrived sometimes and a bit more difficult to debug. But most of them are just defined like this. I don't know if there's questions about this one.
Look, can I ask? I mean, we understand that male longevity with Aedes aegypti is like 2 days, where females is much longer, but you're dealing with gonotropic cycles. Is that factored into this model? I mean, the males don't live very long at all. They're in, they're out, they're gone. It's very different to Aedes albopictus, which males live like a week and a half, but the boys with Aedes aegypti only go a couple of days. Is that factored into the model?
I don't remember if we have that mortality difference. I don't know if you remember, Agassi, because I remember we were mentioning just yesterday. Yeah.
I mean, I don't know what the default parameterization is, but all of that is easily modifiable for whatever need you have. So
Okay.
there are mortality rates built in. You can increase, decrease them based on whatever species you're working with.
And
Yeah,
there's
okay.
one for males and females, right?
Yes.
Yeah.
Yeah.
Okay. Yeah, just making
Okay,
sure.
as long as they're highly differential, because with Egypti, the boys are just hopeless. They're out there and gone, in a couple of days. They don't sugar feed. They just, it's party
Yeah.
and die out.
It's a short, exciting life, I guess.
Well,
Yeah,
I don't know. There's no memoir.
But Nigel, could I just ask you, because Scott always told me that they take 2 days after emergence before they are ready to mate. Is that your understanding? And so is it like 2 days after that initial point of
24
maturation?
hours. My understanding and our, from our work in the field, it's 24 hours. So, and maybe Scott has some additional data that, we don't know, but, it's a 24-hour turnaround, where the genitalia needs to fully rotate and they're ready to mate.
So that makes them 2 days from like emergence of the pupae as your expected lifespan.
2.6. We did some mark release for capture experiments, where we actually, calculated that, in our part of the world, which it wasn't a super tropical place. So, it was a little bit drier than what you might encounter in some other places. But yeah, it was only a couple of days. I think 2.6 days was the mean. And
I just want to raise it because I'm aware that there's these kind of delays in things that often get missed out of models, and like just so but okay, 2.6 days, that's pretty short, but it's
pretty short. But we had for our IIT field trial, we had to release 3 times a week the boys. We had to release the boys 3 times a week to maintain a overflooding ratio that was consistent.
Yeah, cool, thanks.
Okay.
Yeah, I don't know if there is any more questions about this one. Okay. Awesome. So so far we've only been running the deterministic version of MGDrivE. However, we usually use the stochastic version. In this case, I'm gonna run it whilst I explain, because it takes a bit of time. But it runs, MGDrivE handles both systems pretty much the same. The only difference is that we have to set up MGDrivE with stochasticity on, so that it instead of loading the deterministic equations, it loads the differences equations. Some caveats: it takes more time to run, as you can see, because you have to run, well, one trace runs at almost the same speed, but as we need to run more simulations, it usually takes a bit more time, and it generates more files in the system. So that might be another thing to take into account. But the predictions are usually way better and more precise, depending on the scenario. So it's taking a bit of time, but we can see that it already, right? So we can see here we're showing all the traces of the run simulations, and they are all their same results. It's just generating all the traces. In most cases, if you're not approaching the edges of the simulation, let's say fixation thresholds or migration issues, or if you're not doing suppression or things like that, the deterministic is not an inaccurate representation of the process. But in other ones, it might give you strange results. So we usually suggest to use the stochastic version. And we will see in an example with migration why that might be important, especially in spatial scenarios. But it's not difficult to use one or the other in MGDrivE. So that's the takeaway on this one. I think in this one I changed the fitness cost to a different genotype. That's why it's the dynamics look different. But yeah, that's pretty much it. It's honestly just changing a parameter here, and I think the plots function needs to change, but it's all in the demonstrations of the webinar. So just please have a look on those if you need to make some changes. Yeah, I don't know if there's any questions on this one. Oh, yeah, some.
All right. So for the stochasticity, where exactly is the stochasticity added? Do all the parameters become distributions, or the processes, or how is it added?
I would need to look into it again because I can't remember. It's been a while. The mating is one, the migration is one, the egg laying, instead of selecting uniformly from the genotype, it selects, it takes the number of eggs and assigns them to the expected genotypes. The sex selection should be another one. Most of the processes that involve selecting one or the other should be, but on the specifics, I would need to look into it.
Okay.
Yeah, no problem. Other questions? Awesome. Let's see. I think those are the three basic examples. I'm gonna speed up a bit just to allow enough time for Agastya. But I think we're doing good in terms of time anyways. And let's do the Wolbachia one. This one's a bit more interesting because it's not only just Mendelian. Let's run it. I'm gonna run it and then explain in a second. Okay, here we go. I'm releasing way too many, but anyways. The way we set up this.
Can I ask, are we doing, this is a Wolbachia replacement?
It's Wolbachia IIT.
Population replacement.
No, in this case, we're doing the suppression, the incompatible.
Okay, so it's an IIT. Okay, thanks.
Yeah. Well, that's a good question. It wouldn't. So, for example, this would be, well, the code actually doesn't differentiate between those two. The only thing is that we are releasing only males. I think it's in this case for the IIT. But if you released females, it would do the replacement one. So the code is like, it doesn't matter to the code, but we're just releasing males. We can try and release females just in a second. We might try it out just to see how it goes.
Okay.
But yeah.
Okay. I'd love to ask the question because we did this modeling and I don't know the detail of it, but the releasing, the leaking of females, Wolbachia females into the landscape early on is not a problem. But once you start emptying the niche, and if you're leaking Wolbachia females into that landscape, then it becomes problematic.
Yeah. No, yeah, definitely. I mean, that's one thing. If it's the leaking, they're releasing females by accident or things like that, that can be handled with MGDrivE. You just add a probability of releasing females randomly.
Okay, great.
Yep. That would be for the stochastic version. That's one of the things that the deterministic version wouldn't work, but I have an example with the stochastic one, so we will see it in a second. But yeah. So the changes here in terms of the code, everything is remaining almost the same, but we are doing a cube Wolbachia here instead of the cube Mendelian. We are still adding just lifespan reduction, but we are adding a lot of lifespan reduction. We're assuming that the Wolbachia pathogen is having a really high toll on the mosquitoes, but let's ease it out just to have nicer dynamics. Here we go. Oh, actually, I think that's yeah. Well, let's leave it as it was. But yeah, and we're releasing. In this case, it's not as it is not a genotype. I think I might be doing it backwards. Well, in any case. As the Wolbachia is not a genotype, we just have a tag for wilds and a tag for the released mosquitoes. And And that's pretty much it. And let's try and do the female case. Let me release less mosquitoes because this is quite a lot. And it should do the replacement. Yeah, in this case, it's doing the replacement because we released females instead of males. So we are not suppressing, we are just replacing the population, and the seed has a lifespan reduction. The population dips a bit, so it's all still consistent with thought.
Gee, Hector, while we're on this piece of the code, there's a question from Pratima about the releases start, and and you have a parameter for that. So
yeah, the releases start. I think I'm starting them at day 30, but there's no reason. I mean, I just chose it because why not? But let's say we want them to start the first year, and let me just release reduce the number of releases to 1. And hopefully this, instead of starting at day 30 that I was starting it's starting at the first year. And that's pretty much it. It's just a matter of changing this vector of releases. And that's pretty much it. Let me see. In 2 separate drives in the same plot. Oh, I see what you mean. That one would be tricky. So in one simulation, we can only handle one drive. There's no built-in function to handle 2 different drives or 2 different species in MGDrivE at this point. One way to go around it would be to build a cube that contains both drives, because the limitation is not the drive itself, it's the cube. It can only handle one cube. If you built a cube that had both drives in the same thing, like a cube that did both the introduction of the transgene and some other allelic combination did the cleaning up of the drive, it can be done, and we have done it. So that's good as long as you can build a cube. The downside is that if the cube is too large, the computational cost increases significantly. It doesn't scale linearly, so just keep that in mind. And to do the other questions, the release at day 30 and at day 800, it is possible. I'm not going to do it right now because it involves modifying a bit. But for example, here's the way the releases vector gets defined. So for example, let's say we're doing 3 releases. And what's this? The vector looks like this. It takes into account, it takes in the number of released mosquitoes and the time of release. So if you wanted to do a release at day 30, you would just need to modify this to 30. And another release at day 800, you modify this one to 800. And that's pretty much it. It's just a bit of handling the vectors in R. But yeah, it's totally possible. It's nothing too complicated. So, and another question. Yep, no problem. I'll do. And okay, let's do the Wolbachia stochastic. I think it's the next one. I think this one's the same. It's just the stochastic version of the simulation. It will take a couple of seconds. I don't remember if I'm doing anything too different. I don't think so. I think it's mostly the same. So just wait. And another note here is right now I think I'm running 10 simulations or something like that, just because of in the interest of time. We usually run, depending on the drive, we run anywhere, and the type of experiment we're running, we're running anything between 30, 50, or 100 simulations, sometimes 200 if it's too close to the extinction boundaries. But yeah, for now we're just doing 10 just because it takes a while, and I'm on my laptop. But yeah, this is the same thing. We're just setting up the stochasticity on, doing the simulation. Okay. Another point we can make here, we have a variable for the sample time. We usually sample in daily time steps, but if you don't want, if you are limited by a disk space in your computer, you can lower this to be, well, increase it to be every 5 days, and that would save a lot of memory if you don't have a lot of memory in wherever you're running your simulations.
Okay. Can I ask what would be the trade-off of doing that? You know, doing a daily sampling and a weekly sampling.
The simulation would still run in a daily sampling. You can modify it if you scale all the parameters. That's one thing. We have never done it, but it's possible. It's not. I mean, it's doable. The only downside in terms of sampling, well, exporting to disk in a too large time step, is that some of the dynamics, if it's too long, some of the dynamics might be lost. Like when you do the analysis, might be blurred out. It depends on the frequency of the events you need to sample.
Okay.
But for most applications, it would be fine to just do every other day or every week. But yeah.
Okay. No. Thanks for that.
Yeah, no problem. And Summit, please.
Yeah. So just to get an idea of the computational capability, are the simulations you're running right now, are those running across and landscape with many nodes, or is this just a single node?
This is just one single node.
Okay.
I have one that we will see in a couple of minutes that runs in 4 nodes.
Okay.
This one's just running in 1, but it's also running in my laptop, and it's kind of like not.
I see. Okay.
Not new anymore. So yeah, in a laptop it's doable. But if you need to do factorial experiments or large landscapes, that becomes tricky handling a MGDrivE, and the computational limits are reached quickly if you do a large cube and a large landscape, because neither of those scale linearly. So yeah.
Okay, thanks.
Yep, no problem. And let's see. And I mean, all of these examples will be there on the repo, and if anyone has questions later on, please feel free to contact us. So most of these I just set up so that everyone had an idea and a source to download things that are running and tested. So from this point, we're going to run to the linked drives. We're going to do a pretty basic linked drive just with the transient homing into a locus and replacing that one with some probability of replacing correctly, some probability of just breaking the region of the genotype. As I was mentioning, this time, as we also want to test a spatial scenario, I generated a landscape, an artificial landscape. This one's obviously not very natural, but in which we have 4 nodes, in which mosquitoes from node 1 migrate to node 2, then node 2 to node 3, node 3 to 4, and then back. So we have this ring topology, and we'll be doing replacement and suppression, both with the same linked drive. Sorry about that. And yeah, let me close all of this. I'm gonna get it mixed up. And the first one should be replacement deterministic. So let's do replacement deterministic. Okay, this one will look a bit more complicated at first, but it's almost the same structure. Why is it? Oh yeah, it might be. Okay, yeah, I think it's running right. Okay, so now we can see that it's taking more time. As Samit was mentioning, this is 4 nodes, so it will take a bit more time. That's fairly, well, that's expected. And in this case, as I was mentioning, these are the parameters parameterization of the link drive. I'm not going to go into a lot of detail about the parameterization because that has to do with more of the cube itself. But we are adding a fertility cost to the homing allele, to the transgene. In this case, we call it EH. Let's see if it's running. We're also adding fertility cost to the R, to the failed replacement, and to the B, to the broken allele. This is just if the drive didn't home correctly, we are adding another cost there. These ones, if I don't recall correctly, I'm blanking out on these ones. I think they both have to do with the error correction. Let me check in a second. If I don't recall correctly, I think it's when the gene drive replaces, how probable it is to replace correctly in males and how probable it is to replace correctly in females. I would need to check the parameters of here in the cube homing drive. And all of these, for example, can be checked in, let's see, here, linked drive. Jared is the Jared Bennett, one of the former PhD students, is the one who did most of these cubes in the past. And he's extremely good at documenting things. So all of the parameters, he has the documentation on what it should be, which is lovely, just opening the file and going through it. So in this case, for example, the ones I was checking should be here somewhere, but yeah. And these are the dynamic, and this is how we set up our cube in this case. It has extra parameters because it is a more complicated one. If we didn't have values for them, we can leave them blank and it will make the best, it would fit it some baseline parameters that we thought were reasonable at the point we coded it. And for the fertility costs, we have this new vector where the WW of the wild types are the baseline. They don't have any fertility cost. If it's WH, if it has one copy of the transgene, it has one minus the cost associated to the transgene and all of the other combinations. So for example, if it's HH, we're assuming in this case it's additive. So we're having 0.5 fertility cost, but this can be modified as needed and as the data suggests. And the movement matrix, this time it looks like this. Let me just plug it in here. It looks like this. So instead of just having one node, this time we have the probability of mosquitoes staying in that node to be 99,975. And the probability of them moving to the next node in the sequence it's really low, but as it is deterministic, it happens anyways. And this is how we're defining our ring topology in this case. It could be anything, as we mentioned. If, for example, this was a 1, mosquitoes wouldn't be migrating. Or if this had a 0.5 here, most mosquitoes would be migrating from 1 to 4. But it's just how we want to define our landscape. And we're releasing in node 1, I think we're releasing a couple of males. We can see here that the drive is moving from node 1 to node 2 to node 3. And it's driving itself towards homozygous combinations of the transgene. But sometimes, as there's some error rates built into the cube, it is failing sometimes, so we have a baseline error rate in the genotypes, which is expected, so it's all good. I'm gonna set up to run the next example and ask if there are any questions because it's taking a bit of time to run.
Okay.
So yeah.
Can I ask while you're running that, if the breeding sites were mapped out in the landscape, can that be attributed to what you're doing? It appears at the moment it's looking relatively arbitrary about how they're moving. But if you actually had everything mapped out in the landscape, can you contribute that?
Yeah, definitely. We have examples of, for example, we have run these simulations in Yorkie's Knob to test it against the Wolbachia release that took place there.
I know
we have,
yeah.
yeah, exactly, very famous in our field. Yeah. No, we have done it. For example, the way we usually do that is we have scripts to download if the data are available in OpenStreetMaps. We download the footprints of the houses, and we generate from that. We sort of assume that most households contain larval habitats, depending on what we know on the landscape. And from that household's map, we automatically calculate all the distances and whatnot. Just don't think I have an example of them in the repo, but definitely we can. Well, I can send you some videos of how we've done that.
That's fine for 80s. I'm more curious about anomalies in our Australian landscape. It's coastal. We can actually map the breeding sites.
Yeah.
On the coast,
nice.
There, brackish swamps, creeks, things like that. We can pop them in.
That's cool.
Yeah, I'm just wondering how useful that would be in contributing to that spatial structure.
Yeah, no, definitely that would be cool. So, for example, one way to go about it is if you have the GPS coordinates, you can generate a map and calculate the distance with Vincenti or whatnot.
Okay.
And then from that Vincenti distance, you can use a migration kernel that maps the distance to the probability of jumping from one or the other.
Yeah,
that would only take.
Understanding the migration kernel, that's the tricky part.
Yeah. That is definitely the tricky part. And there's the other thing that wouldn't take into account physical barriers. Like, for example, if one node was really close to the other one, but there's a hill or a wall in between, that wouldn't be taken into account. So that would need more tweaking in terms of.
Okay. No, but that's all great. That sounds like it can be placed in the script. Okay.
Yeah. It can be done. It just needs more. It's a bit more manual, I guess, but it can be done.
Okay, great. Thanks.
Yep, no problem. Yes, Summit, please.
Hi. So I don't know what else you guys are going to present, but so you can choose to answer this question later if you're going to get to it. So right now, if I am correct, that this doesn't include anything like the cube doesn't include things like parental effects like Cas9 deposition and effects on like mosaicism and things like that. Would that be something you would add, or do you already have it in a different cube, or would that be added to a cube, or do you need a separate function for that?
Yeah, I mean, this cube, I think it can handle that. So just as a disclaimer, my background in genetics is I'm a computer scientist, so some of my answers in genetics will be weird and will probably not be the most accurate, but we can handle it. It's just this cube is a bit more simple so that it runs in reasonable time, because otherwise it just takes forever. But there are cubes that do handle that one. On the specifics of how and how they were built or how they handle it, we can get in touch later on with someone who knows a bit more, but it can be done. This one's just a simple one so that we can get through them.
And quickly that we've got Rebecca on the line from Tony's lab, and Rodrigo at the moment is doing some very detailed model fits to the constructs that are coming from Rebecca and Tony's lab. It's another case of the delay where the more detailed Cas9 deposition models will make their way into these cubes.
Okay.
Yeah. Thanks. Yeah, no problem. And it already finished, but we can see this. This one's doing the the deterministic version of linked drive population suppression. It is failing because resistance resistance is popping up in the population, but I mean the simulation is running correctly. It's just the suppression. We can see that here it's starting to suppress, but then resistant alleles are popping up, and then we're ending up with mostly resistance alleles in the population. And we can see the delays, how they are spreading across the landscape. So this is one of the ways we can check the simulation. And let's see, gonna run actually, let's run the suppression stochastic one first. Because this is one of the, well, I don't know if it's a suppression or the replacement. Let me check. But this is one of the situations in which it's important to run the stochastic simulation instead of the deterministic one, because yeah, I don't think it's here. Well, it is here, but it looks horrible. So let's just wait for a second. And let's see. Yeah, I don't know if there's any questions in the meantime. Let's see. And yeah, North has a comment that we would handle the parental effects introducing more genotypes to distinguish the individuals that did not have a certain time parent. Yeah, I believe that that's one way to that we usually handle it. That would be more up to John to check confirm. But yeah, I believe that's one way.
Yeah, I guess there can be two phases in sometimes the cube computations. So if it's only the maternal genotype that matters, that can be accounted for within that structure. But there are other intergenerational effects, which actually Jared modeled by doing exactly what Ace is suggesting there, by introducing more genotypes to account for the genetics that the parents of the individual had.
Awesome. And yeah, I don't know if there's any questions in the meantime. This one's taking a bit longer, but it's almost done. Let's see. Yeah,
I have a question.
Yeah, for sure.
Right now, there's no separation in. You don't keep track of somatic cells, right? Like what's happening in the soma. Like, for example, I was thinking things of like leaky expression of Cas9. So Cas9 is expressed in somatic cells a little bit with some probability. Is that would that just be added to the fitness effects? Because I was thinking of combining things like, when you have parental effects and leaky expression, you have to keep track of what was changed by the parental effect, so you can't break something twice. I was just wondering if that is something you guys are adding to any of the cubes.
Yeah, I would kindly defer that one to John, because I'm not entirely sure this one. Yeah, sorry about that. Yeah.
Yeah, I think I mean, when we're doing fits of the lab experiments that coming from Rebecca and Tony's lab and Ethan's lab and Omar's lab, then it's really important to think about the phenotypes. Right. And so mosaicism would come up there, and maybe tying a mosaic phenotype to a cost resulting from leaky expression of the Cas or something like that. Otherwise, I think in this realization of the cube, that would just be captured by a fitness effect, because we're not really looking at the phenotypes, we're really just following the genotypes.
All right.
Look, yeah, can I just add to John's comment? Look, it's very hard to measure this in small cages, and you can really only measure it when you're dealing with fecundity. How many eggs are they delivering to the next generation? Outside of that, it's really hard to measure.
Awesome. Well, the simulation already finished. So as we can see, I think the way I set it up, it deleted the previous plot, but we can see that the dynamics of the population are extremely different from the deterministic version. So this would be one of the cases in which using the deterministic version would probably break down in our analysis. This is due mainly because of a combination of the migration rates being low, so mosquitoes are not diffusing in fractional parts into the landscape. They are choosing to migrate. So the rates at which they are moving would be slower, and the resistant alleles will pop up later. So we have these two combined effects having a different effect on the landscape. And so yeah, definitely suggest to use the stochastic version as much as possible. And I think that's it on my end. I think I'm right on time to pass it on to Agastya for the version 2 demonstration. So I'll take a couple of questions before passing it on if necessary. So anything else that is bugging someone? Oh, let's see, question in the chat. This oh, yeah. So, yeah, in terms of that one, oh, sorry, I'll read it aloud for the recording. Yeah, there's the suggestion of changing the plot colors because it's difficult, and instead of doing the genotypic combinations, doing the allele frequencies. We have another package coded in Python that does all of those things because so for these kinds of quick simulations, it's easy enough to see what's happening, and with cubes that are small, it's easy enough to see what's happening in just a couple of plots. But when the landscapes become large and the cubes are bigger, getting metrics is complicated. Like we have built a separate package in Python that can handle a complex landscape topologies, aggregate nodes in different, let's say, instead of looking at nodes, looking at groups of nodes, I don't know, municipalities, if it was California or regions in the geographies. And it can also aggregate genotypes arbitrarily. So we can handle resistant alleles in one group, transients in another. We can change all of the colors, all of the things. We can code videos or whatnot. But yeah, we do have a separate package. If you're interested, Pratima, I can walk you through how that one works because it's more like data analysis pipelines we've been building here with the help of a lot of undergrads that have helped me out over time. But yeah, these ones are just let's say you run something and you want to check if at least the output makes sense, you can check it here. So yeah. And documentation of the Python package would really help. Yeah, for sure. We have a lot of, it is the one I usually use. I can help out if there's anything. I use it almost every day. So please feel free to reach out if anything's needed on that one. Happy to help. But yeah, that would be it for now. And let's see. Oh yeah, no worries. Not a competitional thing. No worries. I mean, yeah, as this is, I can totally get it. Like, as this is a really interdisciplinary thing, it's really hard to know everything. So yeah, I can totally understand that. So yeah, no problem. We can figure out. So yeah, but this is just mainly to show the package, and we have a separate thing to do all of the hardcore analysis we use for the papers. And yeah, I guess with that said, I'm gonna pass it on to Agastya. So I'm gonna stop sharing my screen, and there you go. I think it's
Great.
good.
Thanks, Hector. Yeah, hi everyone. Thanks for joining. Let's be talking a little bit about the next generation of this software that we've developed and have been working on. I'll be more talking just about its capabilities and its motivation and what our goals with the V2 version of the software is. And I'll just go through a very, very simple example of how to set up a basic simulation. But yeah, a lot of the more complex features do take a bit of time to run, so it's not really well suited for a live demo, but I'm happy to follow up afterwards with more complex examples that you can run on your own. And I'd also encourage people to look at the latest CRAN release of MGDrivE because it has a lot of the new features and it has a lot of example code on how to run them. So let me share my screen. So yeah, I made this slide deck a few months ago just to talk a little bit about the motivation of MGDrivE V2, which was initially developed by Sean Wu in our lab a couple years ago. So the big kind of overarching goal of V2 was to link these entomological outcomes associated with GeneDrive releases to epidemiological outcomes. They can help to fill the gap between kind of cage trial data and what we'd expect from these entomological parameterizations of GeneDrive constructs to expected epidemiological output. And you know, in absence of real field trials of GeneDrive, we can have rough estimates of how these constructs will perform with respect to human health. So one kind of framework we've been using to map these outcomes and parameters is target product profiles, which are kind of just like broad documents that will explore which genetic parameters are most kind of quote unquote important to reaching the epidemiological impacts that we want in the field from these products. So just to talk a little bit about the high level workflow here, we have a bunch of different inputs into our model. We have seasonality profile, the mosquito life history parameters that Hector went over, baseline disease distribution, prior interventions, age structures, and the genetic construct. And all of that gets fed into this whole suite of modeling that will parameterize kind of a single construct in a single location given a single baseline disease distribution and intervention profile. And from that, our goal is to be able to kind of sweep over factorial sets of parameters to understand and then do kind of a sensitivity analysis to understand which of the genetic parameters really impact the outcomes the most. And then our hope is to kind of inform both like the policy side on how field trial developers can use these modeling outputs to understand, you know, how these products will behave in the field in their given location, and also to go back to lab scientists to be like, you know, maybe you should focus on optimizing this genetic parameter because you get more bang for your buck in terms of epidemiological output. So that's the high level workflow there. To just talk a little bit about some of these inputs. So, you know, as we all know, seasonality plays a big part in mosquito breeding patterns and disease transmission. So, you know, our goal is to be able to characterize the seasonality of a potential release site, finding a balance between over-parameterization, such as daily rainfall and temperature data, which is something we have done in the past, but it becomes cumbersome and noisy to really pull a lot of daily data for a long time period, and kind of an oversimplification, which could just be, you know, an uninformative sinusoid. So we are adapting an approach that was developed by Imperial College that takes shape files for a given region, and I believe it can go as kind of granular as you want as long as you have the geospatial data to support it. And it pulls the daily rainfall data from that geospatial region and fits a kind of a mixture of sinusoids using Fourier transform that can capture the general kind of variability in seasonality. And on the right here, I've just pulled two examples, one from Sao Tome and Principe, where, you know, we're developing simulations for, and Mali as well. And we use these profiles in conjunction with previous methods to develop time-varying larval mortality. And that's just one application of seasonality here. We can, any of the rates between states that we've described here can be made time varying. So if there are others that are, very obviously affected by seasonality, we can incorporate that as well. But the first kind of one we've been experimenting with is time varying larval mortality. To move on, we're using the imperial model of malaria transmissions to link to the entomological releases that Hector described. So I'm not going to go through all the equations here, but essentially it's an age-structured partial differential equation model that has humans moving through these various states based on the level of infectivity in the mosquito population and baseline immunity derived from maternal immunity and immunity from exposure and things like that. It's parametrized by only these 3 parameters, which is kind of great because you don't need to really know all of the intricacies of all of these different parameters to get a simulation up and running. But we have the parameter f sub t, which is the proportion of humans who, upon infection, receive treatment, the baseline entomological inoculation rate, which is related to the prevalence in the population through this transformation in the model, and an age structure, which just kind of distributes all the humans in the model according to age. And we discretize the model, so we're not kind of solving partial differential equations directly, but we discretize it into age compartments based on each state. So we use these kind of baseline parameters to characterize, alongside the seasonality, the pre-intervention profile of a given location. So then, when we are developing these target product profiles for a very specific construct in a specific location, we can have the confidence that we used all of the kind of real world data that we can to characterize those locations. Another kind of important aspect of characterizing a location refers to the history of prior interventions that are already kind of existing in there. So right now we're just considering 3 static interventions: the proportion of populations covered by indoor residual spraying, the proportion covered by insecticide treated nets, and the proportion taking artemisinin and base combination therapy. Based on equations developed, in prior literature, these coverages non-linearly modify the mosquito egg-laying rate, the adult mortality, biting rate, and the proportion of humans receiving treatment. So, the more advanced models are kind of characterizing these in terms of time-varying efficacy, wherein, after you implement an intervention, it becomes less efficacious due to resistance or non-compliance or whatever it may be. Right now, it's just a static parameter, and I'll show folks how to set that up in the model. But we're kind of working on integrating that time-varying component because I do think it's important as well. Okay, I'm gonna switch over to the. Oh, actually, sorry, I have one last thing to say about. So one big component of the V2 that was kind of a computational challenge was kind of linking the 2 different models. So the mosquito model described in V2 is formulated as a stochastic Petri net, which honestly I had no idea what that was before coming here. But essentially, it's a computational representation of a continuous time Markov chain. So we allow different mosquito states to move. We allow mosquitoes to move between the different states according to stochastic rates that are determined by the different parameters of those states. So I can show kind of a pictorial representation of how this works. And honestly, it's not super important to understand the details, but just to understand that the basic model is a stochastic model that allows stochasticity between the states on the mosquito side at least, and and all the different rates can be varied based on what the species are, what your location is, and what what the needs of your simulation are. So the tough part was kind of integrating this framework with the imperial model that I described before, because that's kind of a continuous state space partial differential equation model that wasn't really, naturally intuitive to fit into the mosquito model that we already had developed. So we spent a lot of time figuring out how to kind of decouple the two models, and essentially they run in parallel, and in each time step they sample the parameters they need from the other model. So on the human side, it samples the level of infectivity in the mosquitoes at every time step, and on the mosquito side, it samples the level of infectivity in the humans. And we developed this kind of weighted average algorithm to describe the level of infectivity in the mosquito space, depending on the distribution of the gene drive, as well as, the distribution of all the mosquitoes segmented by genotype, because the eventual goal is to be able to model how different genotype profiles block transmission or affect disease transmission. So that's all kind of built in, and I'll show an example of how to parametrize that. But honestly, this stuff is all just going on behind the scenes, and it's not super important. But just to understand that this framework is a bit different to the one that Hector described, which is more in the line of delay differential equations, describing how things move through different states. Are there any questions? I don't have the chat open, so if anyone, awesome. Okay, and then just to talk quickly about the target outcomes that we're considering right now. As I mentioned, the model is age structured, so right now we can estimate numerous outcomes associated with the gene drive release per age category. So on the entomological side, we still have access to all of the different metrics that Hector mentioned, population fractions by genotype, which help us describe how well a gene drive system is spreading through a mosquito population. We may be interested in the proportion of broken or resistant allele formation. Other metrics that we aren't inherent to MGDrivE 2, but have been built into the data analysis pipelines that Hector mentioned. Include things like the amount of time in which the proportion of the gene drive is higher than a given threshold, and things like that, which are more kind of derived metrics that we can calculate. On the epi side, we're able to calculate the pathogen prevalence, which is the proportion of humans in the model harboring the malaria pathogen at any time, and the clinical incidence, which is the number of new humans exhibiting clinical symptoms in a given time frame. So both of these are kind of important to designing interventions and thinking about how a gene drive can can affect and interrupt disease transmission in a given region. And one thing I don't have on these slides that I will, so I'll move on from these slides. And just to show the schematic of the stochastic feature net here. It's basically the same thing as kind of a standard Ross-McDonald model, but here, just instead of formulating each state as kind of a differential equation, we're just thinking of them as random states in which movement between them depends on these transitions, which are the red blocks. And one thing I mentioned, I didn't mention in the slides, but we're also working on incorporating a spatial surveillance system into the model, and that includes the distributing trap nodes. So, Hector mentioned, having a spatial distribution of nodes. Now we're adding a new type of node wherein, if the mosquito does migrate into that node, it can't leave. And then from that, you can kind of discern how, well, there are a lot of really interesting, research questions that you can answer, like when will the first gene drive mosquito reach a certain trap node, or looking at only the trap nodes, can you distinguish how the gene drive is spreading compared to the full simulation? So it gives a little more insight into how, if you were to implement this in the field, how like how distributing traps can be beneficial to understanding how your gene drive is spreading. So that's still in the works. That'll probably be in the next release. But if you're interested in more of that spatial aspect of the gene drive, the gene drive releases like that, we're definitely considering that. And yeah, and basically we have 3 epidemiological models built in, a basic SIS model, an SEIR model, and then this imperial model that I described. So I'm just going to move on to quickly going over an example of what setting up a V2 release looks like. And as I mentioned, like these are a bit more computationally expensive than the V1 releases. So, I would suggest if you're only interested in the entomological outcomes, stick with V1. It's way faster. But right now, these more kind of advanced epi questions are only built into V2. So if that's more of your area of interest, then yeah, I would suggest using V2. Yeah, so just to quickly go through an example here. So we're going to be simulating oh, sorry, there's a question in the chat. Oh, thank you, Sumit. Yeah, we're definitely looking for input on that too. So if you have some thoughts, we'd be happy to collaborate. Right now, it's a very simple formulation where a mosquito. The migration rates, I mean, Hector, correct me if I'm wrong, they've been parameterized from a separate package that Hector developed. But I think we're trying to work on incorporating different types of traps and seeing how they affect migration rates. And then, yeah, once the mosquito's in the trap, it can't leave. But we have functions that can survey how the distribution of mosquitoes in the traps are evolving.
Just a note on that, and I'm gonna plug in on that to our newest project. So we're building a package to optimize the locations of traps, and this package can take into account different variables like the attractiveness of the trap, if it attracts males or females differentially. Let's say it's OV traps, of course, males wouldn't be trapped as often. So we're building that package so that we can both optimize the locations of the trap to catch mosquito, to detect transients in the landscape as quickly as possible. And the advantage of doing it that way is that at the end we generate matrices that are compatible with MGDrivE. So potentially you could generate your traps locations in your landscape, and then just feed it into MGDrivE with a couple of minor changes, but it wouldn't be anything too complicated, so that you can run your simulations within MGDrivE and have the full suite of simulating the transient and be able to optimize your surveillance scenario. So, yep, thanks.
Yep, cool. Thanks, Hector. Yeah, so just to go through this example really quickly, like I mentioned, this is a very basic example. It's honestly not realistic. So like the outcomes that you'll see won't be, super characteristic of any location or anything like that. But just to show you how it would be done, we're going to go through setting up a very basic homing drive replacement system, using the imperial model, and then I'll just show some pretty simple plots of the different outcomes. The more advanced outcomes in terms of prevalence and incidence, especially by age, are not yet built into MGDrivE, and I think we'll probably build them into the data analysis pipelines down the line. So those are still kind of in the works, but will probably be released pretty soon, because it is an important aspect of what we're trying to do. So as Hector mentioned, the first thing we want to set up here is a basic cube. So we're using this homing drive cube, and we can look at the cube. There are many different aspects. I think some interesting ones here are the release type. So we are going to release homozygote males. The genotypes ID is also interesting and shows the genotype distribution of the cube. Here, W is the wild type, H is the transgene of interest, R and B are resistant, functional, and non-functional alleles, respectively. and just the different genotypes there, so we can see those. One thing to mention about the cube and the next part here is that, as Hector mentioned, there are a lot of these are the default set of parameters, but there are a number of parameters related to fitness, and especially on the epidemiological side that can be varied. So there's no issue that if you have other data, and in our simulations, in real simulations, we parameterize these to real data. So these are just the default values, nothing special about them. So the theta here is the epidemiological vector, which is just the default set of parameters for the imperial model. There's a bunch of them. I'm not going to go through all of them, but they all are documented in the vignette. So if you want to look at that, but some of them are related to the immunity. Some of them are related to the simulations, the feeding rates, things like. Yeah, there's just a bunch of different ones. And so, like I mentioned, if there's like, for example, the male and female mortality rates here, as Nigel was mentioning, if you wanted to modify these, all you have to do is just like update the value that you put in here to whatever you want it to be. So super simple. As I mentioned here, our model is age structured. So I've just set up a simple model, setting up the age bins from 0 years of age to 11 months of age, 11 months to 1 year, 1 to 4, etc. With malaria especially, we're often interested in younger ages and how they will react because lack of immunity and things like that. So, if you just wanted to focus on that, you could just set up an age vector to focus more granularly on the ages that you're interested in, depending on, your construct and your outcome. Similarly for older ages, or if, you weren't really interested in age structure at all, you could just set this to like 0 to 100, and that would just cover the whole lifespan. Another parameter that I mentioned here, the percentage of symptomatic cases that get treatment, that's SMT. The intervention parameters, IRS and LLIN coverage, these are just proportions from 0 to 1, setting them at 5%. And then this add interventions function will modify the rates that I mentioned, the feeding rate, mortality rate, and the last one that I'm forgetting, but one of the rates, they get modified using this function. So we set that up. So now getting into the more kind of technical aspects of the stochastic Petri-net framework, the SPN-T and SPN-P are the places in the transition. So the places are the actual states that the mosquitoes can occupy, whether that's egg, larval, pupal, adult, female, susceptible, exposed, infectious. So we just use the cube to set that up per genotype. So if you look at this this object, it's like very, it's a bit complicated, but essentially these before the underscore is the state, and then after the underscore is the genotype. So there's kind of the factorial combination of states and genotypes. Which is kind of a reason why this is more computationally expensive. Basically, we have to consider all of these different states here. So, good question. Oh, thank you. Yeah, sounds good. This will be recorded, and we'll share it with the group. So yeah, and then the transitions are the rates that are associated with the transition from each of the states here. So. Or sorry, they're not the rates. They're just the description of each transition. So, as from a basic modeling framework, susceptible goes to exposed, and exposed goes to infectious, and like all of these different transitions are just described based on the genotypes and what how the model is parameterized. So, as you can see, for a model with I think we have eight genotypes. There's like a lot of different transitions here. Luckily, they're not, you don't need to consider them super carefully. It's just the way the model's set up, and it's all derived from standard mosquito modeling framework. It's just a different way of thinking about it. So this S matrix just gives us the the amount of if you move from one state to the other, just the unit kind of per unit movement. Also not super important, but just you need to have it for the model. And then just to talk a little bit about characterizing the baseline disease prevalence in a given area. So we have the EIR here. You can also, if you don't know your EIR, but you know the pathogen prevalence, there's also a function to convert prevalence to EIR. So I forget what it's called. Let me see if I can find it here. Convert prevalence to EIR. So if you know that your prevalence in your population is 20% or something, you can. Sorry, I'm just reading the chat comment. Oh, interesting. I'm not sure. This might be a question for John, but my sense is that even if the rate of resistant allele generation is low, we still want to consider it because for long time periods, you'll see I can share later on kind of a more realistic simulation. Even if the drive is replaced to a very low level, after like 10 years, you see a bit of a comeback because of the accumulation of resistant alleles. But maybe John, if you had more thoughts on this.
Yeah, I think that that sounds like a good response to me.
And yeah, I wonder, like, we could definitely develop cubes that don't consider those and just have those rates to be zero. If for your use case that's not something you want to consider, then you could by all means kind of develop your own cube that doesn't have those, and just focuses on the genotypes of interest, and it'll be work perfectly fine. Yeah, thanks for the question. Yeah, so like I mentioned, the whole imperial model is parametrized by those parameters that I described above. So we can just calculate the equilibrium prevalence of all of the disease states. You can take a look at this object. There's a lot of different aspects to it, but the one that is important here is the initial conditions of the human state. So you can see all of the different states that I mentioned and how humans are distributed across these states. And just a technical note, like these are all proportions, so all the states will add up to 1. But if you have, there's a parameter in the theta thing in the theta vector, which is which takes in the population of the location. So it's all scaled according to that. By default, it's just set to 1,000. But again, if you're using a real world location and you know the population, you can just set that in there. So now the interesting part is how we model the gene drive effects on blocking disease transmission. And I know I'm just running up on time here, so I'll just run through this quickly. But basically, we have this vector B naught, which tells you the level of infectivity from mosquitoes to humans. So we have this the wild type infectivity, which is basically a probability of if you have been bitten by an infectious mosquito, what's the probability that you'll actually acquire the pathogen. So the wild type we set at 0.59, which is from the literature. But we're modeling here a gene drive where any mosquito with the H allele cannot spread the disease. So you'll see those probabilities are 0. And then in the model itself, when we pull the infectivity from the mosquito proportions, we just take into account the weighted average of the probability and the proportion of mosquitoes harboring each genotype, and then apply that to the force of infection on humans. So that's how we kind of model this this disease blocking ability. And of course, these can all be, again, these are all just examples. You can parameterize this to whatever you want. We have been doing an example where we're using real sporozoite count data to parameterize these B values. So again, it's all kind of very arbitrary and can be parameterized as you wish. And the final thing is just setting up the release vector. So here we're setting up releases from time 0 and for 8 weeks. 8 weeks, we're going to release 50,000 male mosquitoes. And this is just what it looks like here. Again, this is completely arbitrary. You can set it up to whatever you want. These DT parameters are just parameters needed for the simulation. The DT is the daily time step, so 1 day. And the stochastic is how many, basically the rate. So for each movement, we sample from a Poisson distribution. So this goes into the parameterization of that. Not super important. So this is actually running the simulation. I ran it beforehand because it does take a few minutes to run, and I can show the output. But this is all, so everything's kind of set up. So you would need, you would have, you'd be able to run that, and then these kind of auxiliary functions just format things a little better for plotting. As mentioned before, the plots that I'm going to show are just kind of descriptive of what can be done here, but not what we would use for any kind of real use case. But what we normally do is we have a function that instead of writing this to R, we write it to CSV files, and then we use the data analysis pipelines to run. So for this, so here's a plot of the genotype distribution. Again, like not a super realistic example, but just to show that like we're releasing at time 0. So you see the wild type population rapidly crash here and be replaced over time by this homozygote HH population. We have some stochastic variability in the other ones. Again, I think that's just due to the parameterization, but in the real, in the kind of a real world setting, it wouldn't look, as noisy as this. This was only run for 1 year, so we didn't really see the emergence of the R because the rate of R development is pretty low. So you'd have to run it a bit longer to see that. And then just 2 quick plots from the epi side, or just 1 plot from the epi side. So this is the infectious, or the whole kind of epidemiological characterization in the first age compartment. So the way I set that up here was in this tau humans. We set the index to 1, so that just means pick the first age compartment and show us the plots from there. So we do see, these. I mean, sorry that the colors are kind of hard to see, but basically your disease states here, which are the D is diseased, A is asymptomatic, and U is a subpand infection. They all decrease as a function of the gene drive being released, because that, as I mentioned, as the HH genotype takes over the population, those all have associated transmission probabilities of 0. So you see the infection go down. So this is just for one example. In reality, we would want to show the clinical incidence and the prevalence across the entire age population. So those functions we're still building into the data analysis pipeline, but we'll be updating the code with examples of how to use those once those are set up. So yeah, thanks for listening. Sorry that was kind of a whirlwind, but wanted to end on time. Happy to answer a few questions here, or if you email me with any questions, happy to talk through some of the more advanced examples of the V2 setup.
Okay, yeah. Can I ask it, Nigel here? Are you assuming 100% resistance, in your system to the transmission of Plasmodium?
Oh, so yeah, in this example, yes, but we can, like I mentioned, in reality, they're going to be, those values are going to be fit from either lab data or field estimates that wouldn't be perfect transmission blocking.
Okay, thanks.
Yes, Sumit?
I might have missed this when you're describing it, but does it have human movement between?
Not this version yet. We're going to work on adding the epi module to the landscape. Yeah, the tough part is the computational resources. So we kind of have to see if there is a need for that. We do have EPI if you use a basic SIS model. We do have that for network and landscape, but not this imperial model.
Yeah, already with all the things it has, I can imagine that it takes a while to run.
Yeah. Great. Well, yeah, if there's no other questions, I'll pass it back to John.
Yeah, great. Well, thanks a lot, Agastya and Hector. Have we got one more question? Oh, just a thank you. Yeah, thanks, Hector and Agastya. And thanks for everyone for joining. As we mentioned earlier, as software developers, Hector and Agastya, we want to see, we're excited to see people interested in using the software. So definitely do reach out to us if you have questions about using it or interest in collaborations. Yeah, if you do want to get in touch with anyone, everyone's email addresses is on our lab website, marshallab.com. But you can probably find, just by Googling, you'll be able to find that. We'll be recording, we have recorded